Okay, good morning. Here is what we are going to do today. First, we will look at the MATLAB solutions for this manifold learning. So we will first look at example code for Laplacian eigenmap code. And then we will look at a package called the dimensionality reduction toolbox. Dimensionality reduction. In this toolbox, there are many manifold learning related functions. And the second, as we mentioned last lecture, we talked about the tensor decomposition. So here we are going to look at tensor toolbox for MATLAB. There are some basic function in there, so you can know how can you decompose a tensor and using the major components to reconstruct tensor. Then you can maybe think about some of the applications. And third, we are going to discuss some of the principle of cell-cell interaction detection. Okay, cell-to-cell interaction. So the topic I believe we haven't covered in the lecture. We don't have time, or we just don't have this in the syllabus. But now it's become more and more important. Once you have multiple cell types, you want to learn whether there's a ligand receptor mediated cell communications, or how can you use algorithm to detect whether there are certain levels of a cell-cell interaction mediated by this ligand receptor communications. Number 4, we will cover the progress of my spatial transcriptome analysis. The toolbox. So I include more function in there, and we will share and see what we can extend the functionality in there and to perform a series of analysis if you have spatial transcriptome data. Okay, we may take a look how can we download the data from a geodatabase and the process from scratch. Okay, download data from a geo and advanced analysis. So from scratch to high-level analysis. Okay. Let's see whether. Okay, first let's Google MATLAB manifold learning. If you Google, you will have top 2 links which are relevant to what we are going to do. I will first look at the second link. The second link point you to share the source code. It's M function file, and in here, it display. So Kate Taylor, it's a developer and developed this method demonstration and to create this Swiss roll dataset, and then using this nonlinear embedding technique, Laplacian eigenmap, to unwrap it and display it into this two-dimensional space. We can try, for example, using PCA to do it once we have the original data created. So the original data was created online by some source code. Okay. I don't know if you can have access to see this. If you cannot, let me know, because you may have to be a member in the file exchange in order to see the source code. Maybe you, oh, okay, you don't have to be a member. So I think everybody have access to this source code here. So we are going to just copy and paste. For example, I will copy this piece of code and put into my MATLAB. Okay, this is the original data. You have X, Y, Z, 3D. X, Y, Z. So yeah, so that's what our data set is. Okay. All right, so this data set was generated on the in some regular order, so you can have map them with this color. And at the beginning, you have a spectrum of this red color, and then at the end, you have a dark blue, and in the middle, so you have some color to indicate the theory of this the data from beginning to the end. So now we just copy the rest of them. So up to here, you have the construction of this network. And the network, the linker weight was in this W matrix. Okay, all I have to do is just copy everything and put in the in the common window and look at the output. So that's about it. There are some intermediate steps. I believe that trying to try some different parameters. Okay. All right. So next, we are going to click this, come back to the Google search and click the first link. This link points you to MATLAB toolbox for dimensionality reduction by Laurent Manté, and he developed the t-SNE method. Yeah. The original idea is not his, and yeah, to make this, I think the t distribution is his contribution, to use t distribution to at the last step to put the data into this 2D space. Okay, so his toolbox contains many dimensionality reduction algorithms implemented in MATLAB. So it lists 34 of them from the PCA and IsoMap, Laplacian eigenmap, t-SNE, and many others. So what we can do is to click this to download and see how can we run the example code provided by this toolbox. What I'm going to do is just click this, download it to download folder, and then you have to locate it. And this is a zipped file, so you want to use 7-zip to unzip it. I can extract them here. And then you have a tar file, and then this table, you can do another round of extraction. Then you get the folder, so-called DR toolbox. So that's what we are going to look into, and then you double click and you found the function here. Let me know you are ready. So I also copied the link. So in MATLAB, we are going to change our working directory to here. Oh, you have a problem with 7-zip you haven't installed. What you can try is to have a quick installation. From here, you search zip, and you should have this installed. So you can unzip the gzipped file in Windows. So what I have done is to download the toolbox, unzip it, and then I'm going to change my working folder to that toolbox by using the CD and just change the directory to here. So that's what I just finished. Okay, make sure you know how to do it, because you may have to use this to download different packages and then add into your search parts, so you have all the access to all the functions in particular toolbox. But that's a general way you can have those extension of functionality if you need external MATLAB toolboxes. Later, we will download the Tensor toolbox. We will do the exactly same thing and download the toolbox, unzip it. So you need to know where your download folder is, and you change your working folder to here. And then for this particular toolbox, we need to add by right-click this and add this to the parse search. Both the GUI and the techniques. So techniques contains all the function just as a list in his website, and the GUI contains some source code for graphic user interface. In this case, we need both. So I just added, I right-click the folder and add to the parse. So all the function under the subfolders can be called from outside. Yeah, tar file, unzip tar file again. Then you have a folder. Mm-hmm. No problem.
Like, so I got that file,
Mm-hmm.
and then I went to the 7-zip, and then I did.
Yes. Some of your computer you are okay with, yeah. I think you already have this folder here.
And then when I opened it, it just had that.
Yeah, so you do again right here. Yeah. Yeah, just do this. So that's your working folder, right?
Okay.
Double click inside there and copy the whole thing. Yeah. You just copy this. Yeah. So that's your working folder. Yes. Copy and paste this in. Close your string by, so here you have a single quoter, but at the end you also need. Yeah. Okay, so now double click, go into here. That's where, and add these two folders into the parse. Right click, select the folder, and right click. Yeah, go down. Yeah, and next one, go down. Okay, yeah, we are done. So you installed this package. So the unzip is a little bit tricky because if you select unzipped to here or unzip to some folder, so you may not have exactly the same folder name here as I have here, but it's okay as long as you have unzipped, fully unzipped folder. You add and you add these two folders into your search parse, then you are ready to go. So here we just run the dr GUI and by right click and click run. Or you can type this in, and this is a dimension reduction toolbox graphic user interface. Okay, so this interface just allow you to have a quick access by click to all the example data and also algorithm. So the step one, we will select example dataset. Okay, you can have a Swiss roll and as a testing how many data points you want to generate. Or if you have your own, you can have the files load from a text file or Excel spreadsheet, whatever. So we just try to use this Swiss roll example, and I think it's ready, and you can use a scatter plot to take a look. The estimate dimensionality is optional, so I just leave this. Then I can select compute mapping, so that's the access to different algorithms. And let's try PCA first. Okay, and then it shows computed. Then you use scatter plot to look at the 2D embedding by PCA for the original Swiss roll outcome. And then you can select the different algorithms, the isomap, and reduce the dimension. The computed, then still working on that. So okay, so that's the outcome. So yeah, so second algorithm, it will take some time because for this 1000 by 1000, you need to compute the shortest distance and compute the KNN in order to get a geodesic distance. So it's not like a PCA, just click and then get the rotation matrix is done. So you will feel some lagging, the delay of the response. MDS, simple PCA. I don't know the difference between simple PCA and the regular PCA. Let's try simple PCA, not working. Okay, so then this Laplacian eigenmap. Then computed. Oh, okay, this is the results. Kernel PCA. Diffusion map. So I'm just play with this example and try different algorithms, see what's the result look like. If you load another data with the 3D cluster, again 1,000 data points. This example is more like single cell related. And different cluster have different colors associated, so we can take a look at its mapping results. PCA should do a good job here. Yeah. Okay, so you just try and see. Of course, when you ready, you want to go into the source code as function, and maybe you want to look at some of this implementation. Let me find what some examples. So ISO map. So we know the principle of this. Just replace regular Euclidean distance with a geodesic distance by creating a KNN first. Then use MDS to do the embedding. So that's the implementation of this function inside the toolbox. So let's look at first. You see this is found KNN. So you have an input matrix and you have a K. So this KNN matrix will be generated. So the found NN is one of the function inside here. Where is this function here is the build the KNN network. To build a KNN network, you have to compute the pairwise distance first. I believe doing something here. Yeah, I don't know exactly how this KNN is constructed. It seems looks like it's using some technique to accelerate this rather than O by O comparison. But anyway, so in isomap, you first construct the KNN or neighborhood graph, whatever you call it. Then you use the Jastrow algorithm to find the shortest path. Well, for this is you use this distance to replace Euclidean distance. This is so-called geodesic distance based on your KNN network. And then you have these procedures to do MDS. Okay, so this is just similar to PCA, but it just rotate rather than the variables, covariance matrix for between variables, but here it rotate the distance matrix and then project to low dimension. That's all. Okay, so this is implementation for isomap. We also introduced Laplacian eigenmap. So here the function is Laplacian eigen. In this case, we first create a network first. Okay, so we need to build whatever it's graph. Okay, so use either this way or for smaller graph, it uses slightly different technique. L2 distance is Euclidean distance. So once you have the graph, you compute its. So this have using a kernel function to recompute the weight between the link of each link. Then you get the D matrix. This is the degree matrix. So the difference, so here is Laplacian matrix, and they replace some not number into 0. Then with the Laplacian matrix and the D degree matrix, here is to do a generalized eigen decomposition, use L and D. You have option to use different implementation. So here, GDQR is one of the way to do it, or you just use native MATLAB function eigen. The first one, but using the second and above to do the mapping. Okay, so basically that's all. Yeah, on the screen, sometimes you see this warning message. Depends on the situation, and sometimes because implementation problem, there are issues error message. So whatever package and function you may run into, the running errors depends on your input data. So you need to have some capacity. You have the ability to debug or read the source code. So if you want to do it bioinformatics, you really need to have some of those kind of technologies. Okay, we are done with number one. Review some source code for Laplacian eigenmap code, and also look at this toolbox, play with some example data, and then hopefully you learn the habit to look at the source code and read through, and especially when you have a running errors, when you run some source code, you can have a ability to debug and at least to identify where the problem is. For example, here we need to know, okay, that's on the code line 51, and where the error message is, and you trace down, and then at the end, you know, okay, it's a scattered and the function right into the problem. Okay, are we ready to move on to tensor? Okay. So what we can do is to clean also, and also it's better to redirect by click this to download the folder. So because we are going to download a new package and unzip, so you know where you are. I'm going to delete some of these folders here. Yeah, just even before we use any particular tensor toolbox, we can, for example, define a variable. You know, you can create 2D variable. So you can do 3, 6. So this is a matrix. I'm thinking, how can we add one more dimension? So I'm going to create a 3. So this is a 2D. If I do the 3D, I can do so I put this side by side. You see, I can do make it slightly different. So I have this. Okay, basically I put a 2 matrix into another level. To make this more generalized, I can make one more row. Okay, here I'm creating 3D 3D matrix. So this is a tensor. If I can make this work. So now I have A. Oh, well, this is not. It's still 2D. It's still 2D. How can I make 3D? So here I just create a blocker matrix by put some 2x3 matrix in the 4 different corners. What I really want is to add one more layer and put yeah. You're right. So I will redo it. So I will first create this simpler version of it. So I have this. I'm going to put another copy, which to put another layer. So what I can do here is to put so this first layer is A, okay? And then I add another layer for the B, A again. So now I have this B matrix, which have a 2x3, but the 2 different layers. I can add one more and the 3, for example. So now the B becomes the tensor, and you have first layer, second layer, third layer. Okay. For this simple example, is 3 layers has the same matrix A, but I certainly can change. See, for the first layer, I'm going to add 0.5 for each of the element in A. Then I still have this, and this B is 3-dimensional. Okay, so B is 1, 2, 3, something like this. I don't know if there are scatter plot. We can view this. I don't think so. I don't think we have this matrix to view a tensor. We do have image SC. You can view the values using color for A, but there's no way you can do B. Well, I do have a way to view it. I use a subplot. The 2x2 first. Image C, B, first layer. Okay, I do. This should work. So instead of, so I can plot, because I can plot 2D in this color way using different color code. So I can see, okay, I create 3 matrix to show all the values in B. I can do that. So if I do a random number matrix, then this figure will look like a little bit random page of this. Okay, so instead of create B in this way, we can really generate random numbers. For example, this by 2, 3, 3. So this will create a B matrix tensor, 3-dimensional. So this function, like MATLAB, already allow you to go beyond 2 dimension to 3 to higher to create the matrix, to create a tensor. You can add one more. Okay, so this is like a rank 4 tensor. Okay, for example, you have a data set X, okay? The axis, make it bigger. So X is 10 by 20 and 3 layers and 4 entities. I don't know, 4 blockers, something like this. So X is a tensor data. And the majority of functions, just like a PCA, won't work because this function was not built for tensor structure at all. It works fine if you have a 2D data set. So all this function you have used, for example, again, must be a 2D and not ready for the tensor. Maybe SVD, I don't think so. Okay, it must be. So once you go beyond matrix, majority of functions do not work anymore. So either way, you have to deal with this layer by layer, use 2D information, and then incorporate later or process later. but certainly the tensor idea is to integrate all the dimensionality and work on the tensor directly. So that's why you need a specialized package which can take that information and jointly to do the decomposition first, for example, to apply to all the dimensions in the data. So we come back in 5 minutes. I will show you to download the toolbox. So I can shoot. Let's just Google the toolbox. So the Tensor toolbox, if you during the break, you can download and install this. Okay, it's download a zipped file, unzip, and then found the folder. Source code in zip, and then you open by extract here, and so that's the folder. You copy the folder directory name, and then here you cd with a quarter marker inside here, and then we change our folder, working folder, under this toolbox that we are ready. Okay. Okay, let's take 5 minutes break. Come back to look at this toolbox. Okay, here I'm going to create a random tensor. Okay, so this tensor I generated using random generator, and the dimensionality is 10 by 20 by 9. So first, we have a way to show is this 9, the color, using this color map, 3 by 3. So we can visualize this. Visualize what I created. I create a random matrix tensor X and plot nine matrix using this color map, color image, and show its values in this 9 matrix figure. Okay, each of this is 10 by 20. So let me copy this code into chat. make sure you have this. So this data has some problem because there's no structure. We don't see any patterns. So I'm going to make it a little bit. For example, I'm going to create some patch in the first. So let's see, first 1 to 5 row and 1 to 10 column of the first and the first. To make this 0.7. And then we plot that again. So you will see, okay, at least you see some artificial patterns in there. Otherwise, all random. Okay, as I said, SVD will not work in this case directly on X, but it will work on the first matrix, right? So if you have a matrix, it will work. This will decompose it into USV. SVD will decompose this into USV. And USV, the product. Should be the same as original. This has to be transposed maybe. Yeah, so. I hope you understand what I'm doing here. Because SVD can only work on matrix, so I chop out the first dimension from my tensor and gave this matrix to SVD, and SVD decompose it into USV, and USV, when you product them back, and you get the original matrix back. Okay, so you can do this to move to another matrix independently. You can use SVD to do it. And now we will transform this X into tensor. Because you have a tensor toolbox installed, so it's allow you to convert this three-dimensional matrix into tensor, and then their CP alternative authors can decompose this into, for example, 2 components or maybe 3. Should I make 3? Yeah, maybe. So in this way we decompose. Okay, I gave you the source code here. So this is a CP decomposition using alternative this strategy to alternating square error to decompose the tensor by giving number of components. So this decomposes into 3 components. So inside the results M, so this is the output M. M contains U 3 components of the vector collections and for the lambda. So because we ask for 3 components, so it returns 3 columns. If we ask for 2 components, the M will change. So here, if I only have 2 components, then the only 2 lambda value will be given and 2 columns of this U1, U2, U3, because you have 3 dimension, so each of this. Okay, to reconstruct X, let's call XR reconstructed. We can use the function called for and for M. Okay, so this function will take your decomposed tensor components, come back and reconstruct X. So this is reconstruct X. Now we can plot this out instead of using X. Here I will translate into XR to show the reconstructed plot. Well, let me see what's going on here. Oh, XR is not a, yeah, it's a tensor. So I have to change this back to the 3D matrix. Yeah, so this has already been reconstructed from the decomposed components, but it's still in the tensor format. So I have to convert this into matrix. I don't know how to do it. I will try double. So I convert this back to regular matrix so I can plot this. Yeah. Well, let me think about this. Okay. Yeah. So instead of use 2 and 3 and reconstruct double. There are some problems that I may use the wrong function here to reconstruct from the components to the original. So I have to go back and look at the documentation. So once you decompose, how can you reconstruct the original? Because I tried this and my reconstruct matrix looks very different from it's supposed to be, and it's supposed to be like similar to this. Okay, but now it's very different. So I do have to go back to see, okay, where the source code, where I use the function wrongly. I believe it should be this problem here. Okay. Now inside the documentation here, there, if you double click inside the documentation, there are examples using a different data set, which is this provided by the toolbox itself called amino acid. And then I'm not so sure whether we can run this. Okay, we have to add the parse for the TensorBox toolbox into the search parse in order to run this. But anyway, so this is another way you can look at it. So M2C. So you see there are different options when you do this decomposition. Initial values and the tolerance, max number of iterations, and all may have some impact on your outcomes of this results. Okay, in order to run this, you have to go back and add this folder into the search path. And then you can come back here and to run this example code. Okay. It's just decompose the 3D matrix data into components and then also show the values. And those values are generated by using different initial value and setting ups. Okay, so I think we just spend those time for tensor. And then if I can figure out later and the correct way to regenerate the tensor from a decomposed component, I will let you know. I have to read this toolbox more carefully. Next section, we are going to talk about the cell-cell interaction. So I will quickly explain to you what that means here. Okay, so we have a single cell RNA-seq data, so that's input. We have genes. We have cells, right? And cells, if you, let me make this bigger. Now we have genes, we have cells. We have values for those. Okay, some of the genes are ligand. Okay, so here, maybe a ligand. Some genes encoded receptors. receptor. So those just examples. I have one ligand here, and the green is receptor, blue receptor. Okay, for the cells, I can divide cells by cell type. Okay, we have type 1, type 2. In order to detect interactions between one of pairs of ligand receptors, for example, we have database, so we know ligand 1 is paired with receptor 1, and we know ligand 2 paired with receptor 2, etc., etc. So we have all these pairs. We know from the biology, these are the pair relationships. Of course, there one ligand maybe have another receptor 3. It's possible, or same receptor accept different ligand, or one ligand binds to different receptors. So, but anyway, you have this pairwise information. And in our example here, we have one example ligand I with receptor J. So we just generalize this. What the signal you want to find is the expression level of ligand gene in type 1 cell. Okay, so this expression level. So if the expression level is relatively high here, so we have a certain definition of the high or low, whatever. Well, the receptor gene express level is high in type 2 cells. Okay, so this. So let's see, this purple color indicate high expression level. So once you have this relationship, which ligand gene expressed in cell type 1 relatively high, well, its corresponding receptor expressed highly in type 2 cells. If we have this relationship, then we can predict, say, it's because interaction between these two cell types, the activity is strong enough. So it requires the highly expressed gene to support its protein products to be upregulated in order to keep this active interaction between cell type 1 and 2 through the ligand receptor pairs. Okay, so this relationship is directional. So you don't expect the receptor is strong in cell type 1, also strong in cell type 2. Depends on who's the donor, who's the source, who's the receiver cell type. So the relationship is directional. So in this case, it's cell type 1 is the source to provide ligand products. And type 2 cell is receptor expressing cells to receive the signal, not the other way around. Okay, so that's the principle. I hope everybody get the idea here. Well, any questions from online students? So how can we measure the increased level of both expression level of ligand in type 1 and also expression level of receptor in type 2. So here we will look at the cells in type 1 and compute average expression. So we will have expression level of ligand in type 1 cell, okay? So you can take average across all the cells belong to type 1, and then we will multiplicate. So we will times multiplicate the expression of the receptor in cell type 2. So this expression is average expression level across all the cell type 1 and 2 cells in your sample. So if this product is high, it could be either ligand higher, could be receptor higher, but the most likely is to be both ligand and receptor expression is high. Then the product of these two will be higher. Okay. So the cell-cell interaction analysis mostly will just go through all the list of these ligand receptor pairs in the database and look at your data. And you have to decide which cell types, which two cell types you want to do the research for, okay? So you want to study the interaction between these two cell types. So you have to select two cell types first, and then you run through all the ligand receptor pairs in the database and compute the product of the average expression between these two genes in cell type 1 and 2. And then you have to reverse the relationship. For example, the first round you consider type 1 as a donor cells and the type 2 as a recipient cells. Well, the second round we will flip it back and see, okay, I will use type 2 as a donor and type 1 as a recipient because the computation well, let me think. Okay, so it's directional, so you have to run that again. Okay, so that's the principle here, and let's look at examples. For this example, I just switch back my working folder to the toolbox. Single cell gene expression analysis toolbox. If you have trouble, you may have to reinstall using the code here. Yeah, so this have to go Google GA toolbox. So we will still using, we will be using the example data here. You have to be processed because we need annotation of cell types. By the way, this is not a good example because there are a lot of cell, B cell, T cells, so lymph blood cell. The communications between them will not so strong. I don't believe they're. But as example, I just show you the procedures. Then if you have a real data set, then you can use. So in the experimental, we have this selection ligand receptor mediated intercellular crosstalk. This will bring you to this window, and you have to select 2 cell types where you want to study the communications. So I will see select monocyte to T cell maybe. Okay, so the cell type has been selected. Okay, the computation is done, and the program detects some of this interaction. Pairs, so you have a ligand and receptor genes. LK, KL values indicate how strong the interaction is. So the top one is this. Then with this panel, you can click. This panel should still be here. So you see, it's hard to see the direction, but it looks like this TIMP1 to CD63 is both sides. Okay, it's not a one way. Yes, then you can select another one until 3 to see the results. For once you select all the results you want to look at, then you can cancel. That is done. You can move those. So this indicate IL-16 strongly interact with monocytes that's from a T cell to monocyte. Okay, the direction is stronger for this direction, but not the other way around. Well, here to show you expression level of this ligand receptor. You have to refer back to the original annotation, so you know, okay, maybe somewhere these are T cells, and those are the receptor gene expressed in monocyte. Can you see the relative expression level? So on your left panel is this interleukin, IL-16 abundantly expressed in T cells. We know this group may be T cells. Well, the CD4 expression level is relatively high in monocyte. So that's because of this relationship to give you the final results that this direction of the change is stronger. Okay, so this is just one of the examples. Another is this gene, which is stronger between this pair and it's from monocytes to T cell. Okay, this relationship. Look at the expression chart here, and we know this is a monocyte, and you see this ligand gene expressed level is relatively high in this group. Well, in this receptor gene expression level relatively high in the T cell group here. So you see the contrast between these two. Not very clear because you need to take log transformation to get rid of this highly expressed peak to see this, but somehow you can see the one group have a upregulated ligand gene expression, another group have a receiver gene expression. So another example is also from monocytes to T cell. This from this to here. Okay, what do you think is a clear example? Is a clear example. It's better than the previous one. At least here T cell only express this receiver gene, and then so this relationship is not so clear because it's either side. It's okay. Okay. Yeah, the gene have to be expressed at a certain level. If you have, if you have a receiver gene completely shut down in that cell type, you will never able to detect any significant interaction between. So you have to have activities of the receiver gene with or ligand. But it depends on, well, you have to know what pairs you want to look at and which cell type you need to focus on to study. Database. Mm-hmm.
Is there a way to go in and just select, like, I know this ligand is actually receptor, maybe that receptor in my model isn't there?
Isn't there? That's right. So it's a very good question, and some of my users ask for this function. So I want to arbitrarily define the pairs. So I want to look at this first and that first, the compute product for me. I'm trying to build that information in. So right now it's only based on the database provide pairs. A very good suggestion, and I mean the legitimate research question, and we should have that function in there. Yep. Yep. Now, if you can it's better to switch back to the download folder. And then if you can, just click and find a place where you can download the package, but without impact on your other toolboxes. And then to download this spatial transcriptional using the link, the code I just paste in the dialog. If you want to type this in, that's the full command, which is to download this spatial transcription packages. Okay, then you should have this folder in here, and then we're ready. Installed. Okay, ready? Okay, good. So if you are ready, you can double click this folder. So we are inside here. Then we can load example data. Number 5. Okay, then one. Okay, so this is a new interface for spatial transcriptome data analysis. And a lot of this icon, this buttons, the image has to be changed. But for now, you have to move your cursor over the button to know what the function of this button are. Okay, so this is 3D. So you switch this by 2D, 3D, so you know under under this plot, there is this slide section image. This structure here, right? And if you turn this on, so this is called this button here, and you are ready to move your expression pixel. So you use this function to align your expression pixel onto this image, make sure they're aligned very well. So the reason is we are going to sample the image using the location of your pixel, the spots, coordinates, to learn the features from the image. If the image shares the same feature, we are going to cluster those spots into the same group. So it's very important. You have to align your expression spots very well with this image. So you see this image, we have some structure here and this oval and some epithelium, maybe cells in this dark shape and some other structure here. So, so this different tissue type will be learned from these spots by looking at the texture or color of this picture in order to cluster the pixel inside this spots here. Okay, the information will be used. I don't think we have time to do this because the sampling and the training will take some time, but today we may just look at those very basic functions. For instance, if you look at this, it's already been pre-computed clustering algorithm. So you can select, let's see, based on the image analysis, and there is a structure in this orange shape, and then another. So here the clustering. And also you can use SC3, which is single cell expression based analysis. You can get very similar structure of this clustering of the segmentation. So these two clustering are using completely different information. One is from the expression matrix, another is using the image, the pixel. So, but they achieved very similar results. Okay, you can click this button. Say, I want to look at those separated clusters. Okay. So you see this yellow is this one, the purple shape, and the green one is this. So you have a separated, in this case, 6 clusters. You can also click this to select the genes, look at its expression. For instance, I look at expression of genes. This button here. Okay. you see a lot of those genes are not very informative, right? Either expression level is too low and or the distribution have nothing to do with the spatial distribution of either those tissue difference. But this, for example, is okay. So that's why we do need, so if you want to close all the windows, just click this button here. Close all the other windows. So that's why we need a way to select those spatially variable genes. So in this button here, using ANOVA to select some of these genes, which more informative for their distribution. And those are some of the top genes. You can see their distribution. So for instance, this one more highly expressed in this region. Can make this better, I should. I believe last time I showed you how to use this brush tool to select a group of cells. I mean, the pixels or spots here, and then identify marker genes for it. Okay, there are many options you can do with this interface right now. And you can also say, I want to bring up single cell expression tool, and this will just treat all the spots here as individual cells. And your own analysis you have from a single cell expression tool will be available for you to do the analysis here. Okay. All right, I think that's all today we want to share. We are done. Okay, I tried explain you with this principle of cell-cell interaction. So this is done. Now also, okay, I have to show you this import data from Geo. Yeah, but next time, okay. All right, do you have any other questions?
I was getting an error a while back after, or at the beginning of this section, kind of was.
Which part of this one?
Yeah, after I downloaded the spatial transcriptomics folder.
Yeah. Have you loaded example data here?
Yeah, I think I was getting an error when I loaded that because it said that it can't be instantiated as an object.
Are you in the folder here, inside this folder?
Yes.
Yeah, maybe you have some conflict with the previous version because last week we downloaded the same folder. Not so sure.
Okay.
Yeah, but maybe you have a clean download. Maybe try that again. And you
yeah, I'll just delete it and try it again, I guess.
Yeah. Okay, everyone. Then, oh, I see. There's look at this cannot. Yes. Yeah. So okay. So you need to have this installed, and also the single cell gene expression toolbox installed, and also in your search folder. Okay, see you tomorrow. See you next week, everyone.
Okay, is that a toolbox underneath the add-ons, or is it a separate
Yeah.
toolbox?
Yeah, so you
Okay.
need to have a single cell GEA toolbox
Okay.
installed as well, because these two are linked. You have to use that to support this.
Oh, it says it's already installed on mine, actually.
Yeah, what you can try is see if you do CD GEA, can you change your folder to,
Okay.
yeah. If you have this, you should be okay. If you cannot,
Yeah, it worked.
it worked. Yeah, I don't know, but you have to have a clean installation, and also this spatial thing here, and then it should work. Okay.
Okay. I mean, I'll just try it again, I guess, but
Yeah.
the command work, because it did give me the path, and then it switched my path to the folder.
Yeah.
That
So
was
that
downloaded in.
indicates you have that installed correctly. Yeah. And this also, have you updated this first different version? I mean, it could be an old version. Okay, if you don't have any other questions, feel free to log off, and I will see you next Tuesday. Yeah, Alex, you may have an older version of this. You may want to update.
For the single cell gene toolbox.
Yeah.
Okay, it said mine, it's updated from the 7th, so last week.
Oh, from last week, it's supposed to be okay. Okay. Eric, do you have this error message here? No. But you can run this interface or yeah. So Alex, the problem only on your machine right now. We think.
Yeah. I think there was some issue I had when it was unzipping, but I can try that again because I just
Okay.
deleted the folder and I'll try it again.
Okay. Right. See you guys next week. Okay.